This is a production of Cornell University.
It's a real honor to be here at Cornell. When I was a graduate student, I was very much aware of the group led by Ed Lemon here, Bob Musgrave, doing photosynthesis research, sort of moving from the laboratory scale reductionist approach to photosynthesis out into the field. And there's this iconic paper, Son's work in a cornfield, that was maybe 1965, something like that, science. So it's always been, I've always been interested in Cornell. I've never visited here before, so it's been it's good to be here, and it's good to experience the legendary winter in Cornell. I was very lucky to get here, but with Ying's help, we made it and made this work. So I'm interested, as Ying says, in photosynthesis, and what I'm trying to do is to patch together ways to model and measure photosynthesis at large scales, while not forgetting the mechanisms that underlie it. So trying to make the connections. The long-term goal is really to come up with a way of monitoring photosynthesis to understand what controls it, and particularly to see whether things like climate change are changing it, land use change, how are we doing in agriculture and things like this. The photosynthesis is really the primary driver of all these productivity processes, and then of course is linked, as you mentioned, to transpiration and to the hydrologic cycle, and actually to weather. So it's been a lot of fun kind of keeping photosynthesis, trying to keep photosynthesis in the middle of all this stuff, in working with things like Earth system models. So I'll talk mostly today about chlorophyll fluorescence, which Ying is really the expert on, and a little bit about ways that we're trying to build on what we learned from chlorophyll fluorescence to understand the standard multispectral approaches that have been used by the satellites that have been around since the 1980s to see if we can improve the way that they do photosynthesis. Start by, so that I don't forget, these are the folks in the lab who are doing most of the work. This is Grayson Badgley, who's done recently work on reflectance, linking reflectance measurements to the solar-induced fluorescence. Yilu is a modeler, radiation transport modeler. Ari is doing measurements, sort of lab measurements, working out the techniques for measuring fluorescence from fields. Jen is a modeler, working with me on extending the models that we've made of photosynthesis. Lee is doing work on, he's a great statistician, and he's working with us on putting this together to try and do global simulations. Yoichi is doing geostatistics, trying to show how solar-induced fluorescence works as a variable driving or correlating with gross primary productivity, trying to approach it from the data as opposed to the modeling approach. And this is Young-Ril Ryu, who is a sabbatic visitor from Seoul, Korea. And they do most of the work, so I want to make sure they have credit. To start out with, I thought I'd talk a little bit about photosynthesis and the way I look at it. It's driven by the sun, of course, but I think it's useful to think about really how's it linked up. And if you think of it, the radiation is coming from the sun, really comes from the collision of mostly hydrogen atoms in the solar atmosphere right at the top. And when two molecules or two atoms like this collide, there's a really rapid change in velocity. When that happens, electrons jump around, and a good deal of most of the energy of that collision gets transferred into the emission of electromagnetic radiation. And the average energy of those is coupled to the temperature of the sun, which is about 6,000 K. So that's the mechanism that the energy produced by the thermonuclear reactions down in the center get released at the surface. It's just black body radiation. So every photon that comes out is really related to the collision of atoms on the sun. Some of those photons get to Earth, and some fairly large fraction of them actually wind up getting absorbed by chlorophyll if they're in the right wavelengths. It's 8 minutes from the sun to the chlorophyll. An electron is promoted to a higher energy orbital in the chlorophyll molecule. And basically, it has the energy of this collision that happened just shortly ago, and in relativistic terms, simultaneously on the sun. So in a way, a part of that molecule is at 6,000 K, quite hot, could do a lot. It, of course, is unstable in this configuration, but it has trapped, at least momentarily, the energy of an event that's occurred on the sun. And what it's done is essentially promoted that an electron on chlorophyll molecule to a higher state. As you know, the blue absorption of chlorophyll degrades to the red first excited state, and it can decay or move to other decay by fluorescence or photochemistry or other pathways. Basically, these are, what I'm going to use phi for is a fluorescence or a yield, and the yield of all the processes that can be involved in the de-excitation of that absorbed packet of energy adds up to 1. In terms of sort of structural biology, someplace out, this is a diagram of the, or a, kind of a symbolic diagram of the of a photosystem 2 reaction center and antenna complex, where there are 2 reaction centers here in the center, and these are light harvesting chlorophyll proteins, which are the ones that are most likely to capture that energy. The energy is captured and moves from chlorophyll molecule to chlorophyll molecule, and ultimately comes to the reaction center, or something else happens to it. But if it comes to the reaction center, there's a charge separation that occurs, and the energy is captured in an electronic state as opposed to electromagnetic state. So it just kind of diagrammatically, it looks like this. The energy comes in, and the real transformative element in this is 2 chlorophyll molecules that are really pressed close to one another. And when they're farther apart, they can transfer energy as electromagnetic energy from one to another. But when they're really close together, they can interact in a way that where an electron moves from one excited chlorophyll to the other. So it's a charge separation. And when that charge separation occurs, an electron can be provided from water to quench the one that's become positive, and the one that has the electron on it can move that electron over to a quinone and ultimately to a mobile electron transport carrier, which diffuses away. The essence of this is that what started out as an electromagnetic excitation winds up causing an electronic charge separation, which is a kind of a redox reaction. And the energy of that is essentially stored in the product here, a reduced diffusible chemical entity. So what started in the sun with a collision winds up out here in 8 minutes and maybe a few hundred picoseconds as a stable chemical entity, which has enough energy associated with it to fix carbon dioxide and power the biosphere. It's not really fundamentally different than what we do with a silicon photocell. With a photocell, the photoelectric effect in silicon causes charge separations, and these migrate to electrodes, and current flows, which we can sort of kind of store that in a battery, and we can drive around on it. So I tend to look at what's happening with photosynthesis very much in the same way that we're using solar energy to generate or charge a chemical battery. And we can then use what's in that battery, which is in the form of reduced carbon compounds and oxygen, metabolically to go for a run, or industrially to burn fossil fuels to generate electricity or power our cars or whatever. So all of that really goes back to charging this large battery, which is the biosphere. Just by, say, probably one of the best indicators of this is the oxygen in the atmosphere. It takes about 1,500 years for half of the oxygen to be processed by redox reactions that are associated with this battery turning over. So there's, you can see it's pretty active. We kind of tend to think of oxygen as being stable and always there, but in time frames of, say, between the last ice age and now, the atmosphere is probably all new oxygen. So that's kind of why I'm fascinated with photosynthesis. You mentioned the stuff that we did a long time ago in 1980 or so on photosynthesis modeling. This is a picture that I just was at a symposium with. This is Graham Farquhar, Susanna Van Kamera, and me. We did a paper that Graham actually got the Kyoto Prize in part for. So I thought I'd just throw that out because we were all in our 30s or 20s at the time that was done. And it's kind of funny to think that stuff you're doing as a postdoc, actually something sticks and something happens as a result of it. We had no idea that this was going to be important. In fact, we had entirely different goals for doing the work. And it was rejected 3 times, but it's turned out to be reasonable. I think it's the most, it was published in PLANET. It's the most cited paper that PLANET has published, but it took us, I think, 3 years to get it accepted. Just an aside, the whole idea of it is to deal with this problem that photochemistry is coming in at whatever rate the light comes in. So the potential for photochemistry is entirely dependent on clouds and solar angle and things like that, and leaves in between your particular chloroplast and the sun. And the energy is going to be stored ultimately in dark reactions, which result in CO2 fixation and synthesis of sugars, which is dependent upon the availability of CO2 linked to the availability of water, temperature, and the amount of nutrients that are in that the plant has to make enzymes with. So this has an entirely different drummer than this, but there's no place to put excess photochemical products. So you've got to have a way to make this system stay balanced so that the rate at which this occurs and this occurs are balanced, because there's very little capacity for storage in between the two. And that was the very simple idea that led to this paper, that the minimum of the rate of photosynthesis, the rate of photosynthesis is the minimum of the capacity of either one of these reactions. And a lot of the work we're doing right now really focuses back to, well, how does this actually fit together? What is the mechanism that allows this one to go at its maximum capacity until it reaches this one, and then this one to go at its capacity while this one feeds it without large losses between the two? Sorry for the scribbly slide here, but the idea we're working on right now with Jen Johnson is that the real regulation of this is a cytochrome complex that sits between photosystem 1 and photosystem 2. Photosystem 2 is the one that generates oxygen, produces an electron, which moves to the cytochrome, and then moves on to photosystem 1, which lifts it up to the level where it can react in carbon fixation. The interesting thing is that when this system becomes light saturated, this reaction center becomes more reduced, and this reaction center becomes more oxidized. You'd think that both of them would become more reduced, and the reason that it doesn't work that way is that this is kind of a ratchet or a regulator that occurs right in between, and that there is very strong regulation at this point, which prevents, if, for example, you apply too bright a light for photosynthesis, it prevents this one from becoming too reduced and producing really strong reactive reduced substrates. So it just kind of keeps the voltage across this whole system more or less constant while still allowing things to work. So it's kind of a conductor in this, and so I guess I tell you this because we keep working on things like this while we're working on large-scale photosynthesis studies, because they keep exposing things that we didn't understand about the photosynthetic mechanism, and it's kind of fun to go back and play with them. One of the reasons that we got very interested in photosynthesis was related to this yield here, or fluorescence, excuse me. This is the yield of fluorescence, so fractional yield of fluorescence. It's a fairly small component, but it's maybe 1% to 5% of all the photons that come in are re-emitted as fluorescence. The interesting thing is that that's a function of the molecule itself, of chlorophyll. It's like when it's excited, it has a half-time or a half-life, just like a radioactive half-life. It's going to decay just as a function of the time that the excited state exists. So it tells you something about all the rest of these things that are going on, or at least the sum of them. If these didn't exist, then the lifetime of fluorescence would be very long, and you would approach a yield of 1. So it gives you kind of an internal clock to understand how photosynthesis is processing the photons. make sense? Feel free to interrupt, please. So fluorescence is also something that we can measure outside of the chloroplast. So it's, we can then see what's going on in a leaf by just looking at the fluorescence that's coming out of it. And it's been used for 50 years or more to look at photosynthesis. It's one of the really nice things, nice approaches for looking at photosynthesis, particularly in laboratory situations. The other thing that I wanted to point out here is that some of these are passive. That is, there's kind of a wastage term and fluorescence, which have to do simply with a lifetime. And then some of them are dynamic that are essentially physiologically controlled and vary with the system. So we can see how these dynamic components are behaving by following fluorescence. And those dynamic components are photochemistry itself and a regulated step, a regulated competing reaction we call non-photochemical quenching, which is actually kind of like stomates for light. It regulates the amount of light that comes through from the pigment system to the reaction centers. So in essence, that problem of how light is how the light reactions are down-regulated starts before the photons even get to the reaction center. They're quenched by this non-photochemical quenching mechanism. How much to go into this is we can measure fluorescence and we can measure photosynthesis. This is a gas exchange cuvette or system, which can measure the CO2 and water vapor exchange of a plant. And it also has the capability of measuring fluorescence changes in response to changing the light environment. And we can calculate the yield of photochemistry in this system by saying that, well, there's 4 electrons required for every CO2 or 4 photons required for every CO2 that's fixed. And there is we can measure the CO2 exchange, add to it the dark respiration that's going on, correct for the photorespiratory effect, and divide by the total number of quanta, and that gets you this is the number of electrons that are transported to account for the photosynthetic rate that's measured, and this is the number of photons that are absorbed by that system. So that gives you the apparent quantum yield or photochemical yield of that system. With the fluorescence, by looking at the change in fluorescence that you get by applying an intense light, you can get an estimate directly of the photochemistry. It comes about by making the assumption that when you apply an intense light, you essentially reduce all the reaction centers so that there are no photochemistry stops. And fluorescence is going to increase because photochemistry is stopped. These measurements are actually measurements of the yield, not the actual amount of fluorescence. The actual amount of fluorescence goes way up when you hit it with a bright light. But if you use a special technique which uses a modulated fluorescent light, or a modulated light to excite fluorescence, and then you look at only the modulated component of fluorescence, you see that yield effect. Here, the amount of fluorescence increase, that is this variable fluorescence that occurs, relative to the total modulation, the total fluorescence that occurs. This is the dark, this is the fluorescence that occurs in steady state. This is what you get in a bright light. That ratio is actually the photochemistry, the efficiency of photochemistry. And there are ways you can go through and prove that. The interesting thing is that if you turn the light on and allow the system to equilibrate, and then redo this bright light, you don't get near as much fluorescence out, and this ratio is much lower, and that's because the photochemical yield has decreased because the system is responding to the increased light that you've applied to the system. It's downregulated it. The other interesting thing about that is if you look at it from fluorescence or from CO2 exchange, and you just take a bunch of points and calculate the yield of photochemistry from the two approaches, they agree pretty well. So it gives you a pretty strong indication that, hey, you can measure photosynthesis either by looking at the way the photons are processed or by looking at the end result of that processing, CO2. I think I'll just skip over this. The main point of this is that photosynthesis 2 has a whole series of regulatory processes that drive it. Some of them are very quick, some of them take long periods of time, and it's kind of very much like a current regulating IC, responding to things that are happening downstream. We can, as I mentioned, there's this light, stomata for light, that is non-photochemical quenching, and this would be the rate constant for that non-photochemical process, and this the rate constant for the photochemical process. And these data on this side are derived from this experiment here, this normal graph here. So this is the rate of CO2 uptake, and this is the concentration of CO2. And of course, as you increase the CO2, it goes up and up to a fairly high rate, and then it saturates at CO2 saturation. Here, you could actually improve this rate by giving it more light. Here, you can't. It's limited by the capacity of the CO2 fixing enzyme, which is CO2 dependent. So out here at a high rate, this is the rate of photosynthesis. So this is this point and this point. This here and this here correspond to one another. Photochemistry is the rate constant for photochemistry is really high. The rate constant for non-photochemical quenching is really low. And as this begins to go down in photosynthetic rate, this non-photochemical quenching turns on, and it corresponds to fairly stable region where the photochemical rate constant stays the same or about the same. So the system is responding in such a way that the light coming in or the photons getting into the reaction centers are not, they're being reduced. And the rate constant for photochemistry is staying about the same. And then that system saturates ultimately, and the rate constant for photochemistry also drops as the reaction centers close. So it just to give you an idea how that teeter-totter kind of thing that I illustrated to begin with works. And this is, there's a lot of power in you doing fluorescence. But I guess the main thing that I point out here is that this PAM fluorimetry, the approach that we're using to make all these inferences about the way photons are processed, requires that we manipulate the light. And that's not on if we're looking at the planet or even a field. As we get out into the field, that's just not possible. So we had this great method, but we were very frustrated with the idea that, hey, we're never going to really apply this in the field. We did some things like put together laser excitation systems to try and pull this out. But you actually can't make a laser that is in the visible range that's any brighter than a pointer. And in fact, if you actually went back and tried to license with the laser licensing regulations one of these pointers, you probably have to reduce most of their outputs considerably. these are kind of grandfathered in. So there's not much we can do to go out in the field and provide bright light to manipulate. So we'd more or less given up that it couldn't be used for regional or global scales, even though it's such a powerful technique to look at photosynthesis with. And that all changed in about 2010 for me. I was walking around a meeting, the poster session of a meeting, kind of at the close of the meeting, looking at posters that had nothing to do with me. And I stumbled on this poster that said, hey, we can measure fluorescence from space. And you know, I wrote down the name and got on the phone the next day. It turned out that it wasn't actually such a new deal. It had been measured before. The technique was originally worked out in the 70s using very narrow filters to look at the light that comes off of vegetated surfaces. It had actually been measured with a reflectance satellite. One of the things that's kind of interesting about this comparison here, these are crop circles, irrigated patches in the desert. And you can see these are in RGB color, and these are in fluorescence. And this is really dark. There's no light coming off the soil. That is, the light that's coming here is the light that's coming from chlorophyll fluorescing. And one of the things that bedevils remote sensing in general by reflectance is that there's an awful lot of light reflected by the uninteresting part of the land surface. And it really messes things up. And here you can see that, forget it, you don't see it at all. If you could just look at that, you'd see the plants much more specifically. And that was the punchline from these two papers that simultaneously figured out how to acquire the fluorescence from satellites that were originally designed to measure the concentration of gases in the atmosphere. And this is a year's fluorescence measurements showing that areas like the Amazon have and the Indonesia and tropical Africa have the highest rates of fluorescence. And things more or less follow the patterns that we expect for photosynthesis. So even though we couldn't do any experiments on manipulating the light, it looked like this is coming out that's looking kind of like we hoped fluorescence would look anyway. And this is a graph from a recent paper that Ying is the senior author on, looking at fluorescence from a satellite that was originally put in orbit to measure carbon dioxide concentration, and flux measured at CO2 flux towers simultaneously with the fluorescence measurements. They agree pretty well. So this is pulling it down to a smaller scale. The other was really based on what we expected for photosynthesis. This is really based on observations of photosynthesis. So it's getting close to home. And if I can make this run here, this is kind of fun. This is a yearly fluorescence changes in fluorescence. And if you look we look at it, let's go back. If I can make this repeat. Call your attention to the corn belt here. You can see that that's really bright. It really comes up. It's actually the brightest spot on the planet. Brightest large spot on the planet. There's other areas that look almost as bright. The Ganges Delta area, some parts of Europe. That's what that's saying is that photosynthesis in the Corn Belt, probably during the time of peak crop production, is the highest of any place on Earth in at least a large a large patch. This is just one of the things that we try to do is to measure this also from field instruments. This is a fluorescence equivalent of a satellite sitting above a soybean field that we're working on this year. What we're trying to do is this is the absorption spectrum of chlorophyll, the green, and the red is the emission spectrum of chlorophyll fluorescence. And what we're, so light that's absorbed here comes back, some of it comes back out in these bands. And, you know, if we were doing this in a laboratory, we just put in a filter so that we only use light, we only light, the plant would see light that was in this range, and then it'd be dark in this range, and we could see the fluorescence from that. Very simple. That's kind of hard to do, again, in the field because filtering the sunlight doesn't work very well. So it would require a pretty large filter to filter the Earth's input radiation so we could look at fluorescence. But the way that they figured out how to do this was to look at the spectral composition of the light that's coming down. That, of course, affects the spectral composition of the light that's going up. And what's shown here are a lot of what looks like noise, but it isn't noise. It's changes in the composition of the intensity of the light as a function of wavelength, which occurs because of things like iron and magnesium and elements like that in the solar atmosphere that absorb light. And they cause these little divots. And you see them again in the upwelling light. Because it's essentially sunlight, it's reflecting, it doesn't change its spectral composition. But fluorescence is new light. There's no impact of the there's no impact of these spectral features of the sunlight on the spectrum of the fluorescence. So if you think of it, the light that you get back out is a function of the fluorescence coming out that doesn't have any of these divots in it, and the light that's coming from the sun, which does. And if you use a statistical tool, it's kind of like n-member unmixing, you can separate those two. You can determine what fraction of the total near-infrared light that's coming back at you has come from fluorescence and what is just scattered light. And that is made possible by having satellites that have the spectral resolution to resolve these lines. We focus mostly on these lines, which are due to Fraunhofer's Fraunhofer absorptions in the solar atmosphere. This big feature is due to oxygen in the Earth's atmosphere, and a lot of people work with this. It's a much bigger signal. It's much easier to separate fluorescence from scattered light using this. But unfortunately, it's also due to the oxygen in the atmosphere. So what you see depends upon how much of the atmosphere is between you and your measurement and what's happened to the light as it's come through the atmosphere before you actually do your measurement. So it's a more complicated thing, but it's possible, and this is the way that we make this this determination of how much fluorescence there is. This is always a bit of a problematic area, so if you've got any questions about this, feel free to interrupt and ask. Does it make sense? Okay. This is just, if we look at fluorescence coming off this is from an Amazon canopy, and all of this is due to clouds. But if we look at the fluorescence that's coming out, it's fairly well behaved, and it behaves pretty well. If we look at, there are ways that we can sort of normalize this either to the incoming light intensity or to the amount of light that's reflected. So it's kind of well behaved. The measurement is difficult if we're looking at these very small spectral features, but if we're really careful the way we do spectroscopy, control temperatures, do a lot of signal averaging and things like that, we get pretty good data. So yeah, we got this method based on resolving these fine spectral details that gives us the light emitted by chlorophyll. It's a pretty powerful proxy for photosynthesis. And I guess the initial thought that we had was that this would all be due to changes in fluorescence yield, which would tell us about the physiology of the plants, and that would tell us all about photosynthesis. As we've gotten more and more involved in this, it's become clear that the structure of the canopy that's doing photosynthesis has an awful lot to do with how much fluorescence gets out, maybe even more than the yield. So, and that has the really key component here is what we call the escape probability, which is the probability that an emitted fluorescent photon that's emitted in the canopy actually makes it out so that we could measure it. And that's very dependent upon both the observational geometry. So it changes as say a satellite looks at the surface from different directions, or if you're looking at the same piece of canopy and the sun were to go through the daily course, you'd also see that this emission parameter would change. So it's a directional property of the canopy itself. Just to sort of go back to that diagram, what's happening here is that a photon is absorbed and some of it gets to the reaction center. Some of it, since this takes a finite period of time, chlorophyll fluorescence comes out while it's in transit, and it tells you essentially something about the lifetime that the photon or the excited state has in this pigment bed. Longer the lifetime, the more fluorescence is going to come out relative to other things. I think I probably skipped all this, and just say that we have we've been working to, of course, one thing to do is to put this into a model of photosynthesis and see if we can figure out, well, how's physiology control this? And this is a series of simulations where we're looking at photosynthetic rate as a function of light. If we change the amount of the CO2 fixing enzyme Rubisco, so here's very low levels and very high levels, this has an effect on the yield of photochemistry. We've got more capacity, the yield of photochemistry can be higher. And you get some fairly complicated patterns of the fluorescence yield as a function of decreasing the Vmax. And this here is the simulated solar-induced fluorescence. You can see that there are some differences. As at high photosynthetic capacity, you tend to get more solar-induced fluorescence than at low photosynthetic capacity, but it's nowhere near the linear zero-intercept relationship which we see in Ying's data, for example. If we do simulations with a model that incorporates radiation transport, photosynthesis, and fluorescence yield, and we vary things which vary the near-infrared reflectance of the canopy by changing leaf angle distribution, leaf area index, fractional vegetation cover, soil color, and things like that. We change the amount of solar-induced fluorescence, and if we go through that same matrix of things and change the fluorescence yield, you get a different slope. So it looks like what we see is something we see two effects. One is related to essentially the structure of the canopy, and the other is related to the fluorescence. And we have the two things simultaneously affecting what we see from looking from space. This is just to show you something about what we expect from the fluorescence itself based on those models. So we're looking with a satellite at the same time of day. Most of the satellites that we're using have polar orbits and they come over at the same solar time for each space. So we're always looking at the same illumination. This is, say, illuminated with full sun and lots of photosynthetic capacity. This is the photochemistry. This is the non-photochemical quenching. This is the sort of wastage term, and this is what's lost by fluorescence. As you go to lower capacity, the photochemistry goes down, the non-photochemical quenching goes up, really goes up when you have stress. But this is the fluorescence, and this is that fluorescence expanded 10 times. So this just shows that, yeah, there's some sensitivity to the environment, but that sensitivity is not the whole story, at least. We have wanted to do this to couple with these kinds of measurements. You can measure the gas exchange of a system. How are we doing for time? You can measure the CO2 exchange and essentially reconstruct what the gross primary productivity is, but these are areas that are about 1 kilometer at most in size. And the solar-induced fluorescence measurements that we're trying to make are normally in the range of, well, the most frequent one, the one they use the most, has a footprint of 40 kilometers on the side. So they're too big to actually match with these sites. But if we could match with these sites, we have maybe 100 of these around the world, maybe 1 kilometer squared each. That's at least a sampling of 1 part per million of the biosphere. And that would be useful in putting together some information about how the whole biosphere works. I think you understand the limitations of that as well. But probably the best approach that's been made so far to do this is to use machine learning to analyze these. What student in our group, Yuichi, has been doing is to look at gas exchange in fairly large scales, air moving across the continent, and try and use models and data to predict or to explain the variation in the CO2 concentration. If you know what the CO2 is at start and you know what at the end, you should be able to say something about what CO2 exchange has occurred over the path that it took across the continent. The interesting thing from this is just that the models that we have that are sort of the state-of-the-art Earth system models get anywhere, the best one does 0.6 or 0.4 of the variance, and most of them are down here in fairly low part of the variance. Solar-induced fluorescence by itself is a single variable without putting in the meteorology and a bunch of other stuff which are in these models explains more. You can explain more of it by putting in a couple of other variables, but it's cool that this is something that seems to capture a lot of the variance that occurs. And what you want to do when you're making a model is put in the data that's has the largest leverage and ignore the data that doesn't have any leverage. And models, principally, we don't do that when we make models. We put in everything, including the kitchen sink. So this is kind of a refreshing way of looking at it. So I think we'll skip over that quickly and go on. The idea that we worked on was the observation that the light that was coming from reflected by the canopy in the near infrared seemed to correlate quite well with that that was coming off fluorescence if we were looking at full canopies. And this is sorting out pixels by their normalized difference vegetation index. So when that vegetation index is high, you get a really high correlation with the near infrared reflectance. And as it goes down to less vegetated pixels, of course, that correlation goes away. So we thought, well, let's just multiply the total near infrared reflectance times the NDVI and see what happens. And this is SIF and that product. They fall in line really nicely. And the thing that's really nice about this, even though it's kind of a derived product, we've got that at 300 meters resolution, 300 meters ground resolution, and we have it from 1980 to present. So we can now look at flux sites with something that looks a little bit like solar-induced fluorescence. It isn't exactly the same, but it has some of the same properties. This is just looking at this that correlated with some global models. Again, just the annual NIRV sum and the GPP modeled. So it captures a lot of the variability in the biosphere all by itself. What we're looking at is essentially the normalized difference vegetation index is this reflectance here. This green is the reflectance of a leaf. And you look at it, it looks at the ratio of this band 2 and band 4. The difference between those two over the sum of those. And this is a spectrum of soil. You can see that there's some slope there, but not nearly as much. So that that vegetation index for the soil, if you were looking at the soil, would be very low. The vegetation index, if you're looking at a vegetated area, would be very high. And if you kind of think of it, the light that's coming off of, say, a mixed pixel like this, you have a very low NDVI of the light that's coming up from the soil. But if that's coming off from a leaf, it has a very high vegetation index. If you multiply the total times this ratio, the NDVI, this index, the NDVI, you essentially get something, if it's all coming from the leaf, it's going to be high, and the near infrared that's coming off of the canopy is going to be all from plant. And it's going to vary pretty much by what the fraction of light that's coming out of the system is that's coming from the leaf. It's actually analogous to the way that we're measuring fluorescence. We're just determining how much of the near infrared light is coming off leaves and multiplying that times the total. And that gives us an index that's kind of like the fluorescence. And this is what you see if we just take the total NIR and compare it. This is for pixels. So this is actually for all the land surface. So this is just the total near infrared coming out, and you can see it's scattered. There's some indication that there's something there, but if you just multiply it by the NDVI, you get this nice correlation because it throws away all these pixels that are influenced by clouds or soil or other things that interfere. And if you just take these are flux sites now. If you take the, say 100 or so flux sites and look at the annual GPP sum and the sum of this NIRV, some other things that we can analyze, it's actually quite an improvement over other approaches that have been used in the past. And one of the things that really we found very interesting of this is that if we took this NIRV, you would think that if you took that and multiplied it by the light, it'd improve things. But it actually makes it worse. And we think that what's going on, what we're finding, is that there isn't, the NARV has most of the environmental correlates that have normally thought to drive photosynthesis already kind of worked into it. And when you add another one, or add one that's already in it, you just add the noise. We're very interested in it from that point of view, because that means we have the system is growing a canopy which has some, I think we'll just skip over these, except to show you that canopies are very diverse. Plants grow those canopies under genetic and environmental control. They have very different properties in the way they transport radiation, but the way that they reflect near-infrared radiation seems to have a lot of information about how they actually are functioning. And we're really interested in it from the point of view of understanding the biology of this control. So I'll stop first, before stopping, just mention that there are a couple of other things that are really interesting in this. If this is an area where we're looking at an area in the Amazon with two different remote sensing products. One's better cloud filtered than the other. The better remote sensing product has a tighter correlation, so we can improve this by getting better remote sensing. This is a C3 soybean crop. This is a corn crop. Same you can see that there's a big difference, as you'd expect, because of C4 photosynthesis. Things like that can also be put in to improve this system. It works well monthly and does as well as the best models all by itself, and does a pretty good job of simulating global photosynthesis, and sum it up there. So thank you very much.
Questions?
Yeah.
One quick question. So what about the oceans? Can you see the oceans?
Yeah, actually the oceans are easier because there's no substantial reflectance in the near infrared. So they've been using it for some time to do fluorescence. Most of the work has been done looking at things like nutrient limitation. There's a new satellite that has just come online called Tropomi that allows you to do red fluorescence. Red light gets out of the ocean. The near infrared light that we're working with, the ocean is black in that range. So you can see coastal blooms and things like that. We haven't worked very much with that at this point, but I think it's going to be useful.
Yes.
I'm curious if there's any effect of the polarization of skylight that changes during the day on your measurements.
Yeah, well, certainly the reflectance remote sensing there have been satellites put up to look at polarization of the light. There's also an effect. I mean, the specular reflectance off of the leaves is polarized. So, you know, if light strikes the epidermis and then is reflected back like a mirror, that's for reflectance a real problem because it doesn't carry the signal of the vegetation. And they've been able to eliminate that by looking at the polarization. There is also the satellite that measures the OCO2 satellite. It changes its orientation as it goes through from north to south, from pole to pole. Because of polarization, and I don't understand the spectroscopy of that. Maybe you do, but they do that. They rotate the position of the slit because of the polarization effect. So it is important, but I don't understand. Yeah.
If you're dealing with a region that has a water limitation, drought or semi-arid, and stomates closed, CO2 entry into the leaf is limiting photosynthesis, how does your system operate or deal with that?
Well, if the we don't have an awful lot of measurements where we have regulated control of water availability at this point. We have drought episodes. What we see is that droughts almost invariably result in lower fluorescence. It could be we see that droughted leaves have less fluorescence, but not a huge effect. It could be that there is an additional effect due to leaf dropping, leaf curling, leaf abscission, and things like that that happen consistent or simultaneously with the drought, which, interesting, also has to do with just shedding light, just as the non-photochemical quenching does. The plants are responding to a drought by absorbing less light and letting less of that light get to the reaction centers, because because of the limitation of water, become less able to use that light for photochemistry. Yeah.
So I'm struck by the intensity in the Corn Belt in the movie you showed.
Yeah.
And what I'm wondering is that because of the agriculture that's there, or is it because of the location? So if we didn't have crops there, would it be just as intense?
It's a really good question. What you tend to see, and we'd have to look for small areas that are, I think there are some sites that are in the Corn Belt, but in forests, for example, that we could look at and figure that out. My expectation is that the whole thing's pretty much limited by the season and the environment, but that for crops, the productivity is squished into a very narrow window, and for forests, it kind of gets spread out, and I wouldn't be surprised if they tend to be more the same if you add it all up over the year, but I don't have a good answer for that at this point. Yeah.
Did you use this technique to look for chlorophyll on other planets?
Yeah, we've you see a very substantial change in the reflectance of the planet in these visible wavelengths, visible and near infrared wavelengths. It's been suggested as a way of looking for photosynthetic pigments on other planets. I think the more likely way that we'll get to that in the near future is looking at the light that gets through the atmosphere as the planet transits across the star. But yeah, I think it's worth a shot. Certainly worth thinking about.
Any other questions? Yes.
What do you mean by using a spherical final distribution in drawing?
That's a good point. What we do in almost all of our models of plant of radiation transport, for example, Earth system models, is we have all kinds of very elaborate descriptions of the water relations and the physiology of the plant, and then we put in an arbitrary function for the way that light is absorbed by the leaves, and that can be spherical or rectifiable But people have avoided dealing with the explicit radiative transport of the canopies. And our work is saying that that is really important, and we aren't going to get photosynthesis right until we do that, and understand how that relates to how enzymatic capacity is also distributed among those leaves. And the other thing that's, I think, really interesting about this is that the work kind of suggests that photosynthesis isn't that strongly limited by physiology. It's more limited by the amount of canopy and the structure of the canopy that's made. And we're not dealing with that at all in modeling in modeling the Earth system at this point. So that's what I mean by it's wrong. It's wrong.
Any other questions? Oh, yes.
Have you made any, you may just say no, but have you made any tacit assumptions about the nature of the photon itself, or the nature of light itself in coming up with your models?
Well, probably we have, but and this whole thing about the thing on the sun and the linking to the chlorophyll, that sort of thing, is something I made up just recently. And I've cornered a couple of physicists to ask them, you know, is this really off the wall, or is it about the way to explain it? And they said, yeah, I don't like the vocabulary you're using, but it seems about right. But if it would be, yeah, we should.
I would like to give you one.
Okay. Yeah, good.
That's Joel again.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.